Hi everyone, this is Ophelia Ibanez, and we're going to be talking about our first topic, cell physiology, functional organization of the nervous system, membrane physiology, nerve, and muscle. This first topic will be subdivided into 3 subtopics, and they will have their own corresponding learning objectives. We're going to proceed with our first subtopic, cell physiology and cell membrane physiology. I need you guys to pause this part of the video and read the learning objectives so you understand what you should be getting out from this session, okay? Now, when you're done, we're going to proceed to our topic outline. So this is essentially how we will be discussing our first subtopic. So you can also pause, take time to read and digest, and when you're ready, we're going to proceed. Alright, the functional organization of the human body. For this slide, we are going to be talking about the cell. So the cell is the basic living unit of our bodies. It provides the structure for the body's tissues and organs, and it also uptakes nutrients and converts them to energy. Essentially, they are our building blocks, and they also contain our body's hereditary code that controls substances produced by cells, of which are essential for the duplication of our cells. Alright, now this time we're going to be talking about the internal environment of our body. So, in our bodies, we have 100% total of body fluids. 60% of that is water and 40% are other fluids. So essentially, if you think about it, we are composed of 60% of water. Within that 60%, it is divided into 40% of intracellular fluid and 20% of extracellular fluid. Now, that extracellular fluid is further subdivided into 15% of interstitial fluid and 5% plasma. Moving forward, we are now going to talk about our intracellular fluid, or our ICF. So our ICF, this is found within the cell. So if you see the picture right here, this is our cell. So everything inside that cell, that is called your ICF. Alright, so this is where most of the fluid in the body is found. Now, this is what I want you guys to remember. Take note of the ions that are part or that are found in your ICF. So these are your potassium, your magnesium, your phosphate, and your protein. So these ions or these compounds are the most abundant in your ICF. So your potassium, all right, and then magnesium, phosphate, and proteins. Okay? Next, our extracellular fluid, or ECF. Now, our ECF, this is 1/3 of the fluid that is found in the spaces between our cells. So everything, these are our cells, all right? Now, everything found between those cells, that is your ECF, okay? So our ECF is in constant motion throughout the body, and this is transported rapidly in circulating blood. Now, ions and nutrients are found in the extracellular fluid, of which are needed by cells to maintain life. Okay, so if our ICF had potassium, magnesium, phosphate, and proteins, our ECF naman, the most abundant found in our ECF is our sodium, chloride, bicarbonate ions, glucose, fatty acids, amino acids, carbon dioxide, and cellular waste. Alright, so for our ECF, it is our sodium, chloride, bicarbonate ions, glucose, fatty acids, amino acids, carbon dioxide, and cellular wastes. Now we are going to talk about homeostasis. So homeostasis is the maintenance of nearly constant conditions in the internal environment of the body. Okay, so all our organs, all our tissues in our bodies, they contribute to maintaining homeostasis. So even in the presence of a disease, our body, or there are homeostatic mechanisms of which will continue to operate in our body so that it will stabilize the body's functions through a lot of compensations. Let's try to ask ourselves, how does homeostasis happen? So homeostasis happens through control mechanisms that control intracellular and extracellular functions and control systems between organs. Okay, so this is why we are talking about our ICF, our ECF, and the cells. Okay, so these control mechanisms are usually negative feedback loops. So that's why we have the picture here. So this is an example of a negative feedback loop. So for example, the relationship of the respiratory system with the nervous system that is regulating the concentration of carbon dioxide in the ECF. Now, if let's say our carbon dioxide is too high, the body will want to lower carbon dioxide inside the body, or lower the carbon dioxide concentration. Now, that's what we mean by negative feedback, okay? So then, what the body will do as a compensation is it will increase pulmonary ventilation. All right? Another example is our pancreas regulating glucose in the ECF, and our kidneys regulating concentrations of H or hydrogen, sodium, potassium, phosphate, and other ions in the ECF. Now we're going to talk about the cell and its functions. Okay, so the picture right here, this shows a picture of a cell and the different parts of the cell, which we will be talking about later on. Let's start first with the organization of a cell. Now, in a cell, there are two major parts. Number one is your nucleus. So your nucleus is this purple thing right here. And then the other is your cytoplasm. Okay? Now, your nucleus is separated from your cytoplasm by a nuclear membrane or a nuclear envelope. This right here. Okay, to separate that from the cytoplasm. And your cytoplasm is separated from the ECF outside by the plasma membrane or the cell membrane. Let's now talk about the protoplasm. So what is our protoplasm? This is the collective term for the different substances that make up a cell. Your protoplasm includes your cytoplasm, your nucleus, and the organelles that are found within your cytoplasm. Okay, now it has 5 basic substances. We have water, okay, which is the principal fluid medium of the cell. So this is about 70 to 85% of the cell. And we have several ions, of which are your potassium, your magnesium, your phosphate, your sulfate, bicarbonate, smaller quantities of sodium, chloride, and calcium. Now, these ions provide inorganic materials for cellular reactions. We also have proteins. So proteins make up about 10 to 20% of the cell mass. We have different types of proteins. We would have, one is your structural proteins, and the other are your functional enzymes or functional proteins. Okay? So your structural proteins, these form microtubules, which are the cytoskeleton. So cytoskeleton of your cell. And your functional proteins are the enzymes of the cell and are mobile in the cell fluid. Okay? Next, we have your carbohydrates. Your carbohydrates provide nutrition for the cell. Glucose is always present in the ECF. Okay? And glycogen is stored for later use. Finally, we have your lipids. So your lipids, these are insoluble in water and are therefore used to form cell and intracellular membrane. So these are barriers that allows for separation of the different cell compartments. Now we're going to be talking about the physical structure and organelles of the cell. First, we have the membranous structures of the cell. Remember, we talked about lipids. So the membranous structures of the cell, these are composed of lipids and proteins that act as barriers to prevent water-to-water soluble substances from moving to one cell compartment to another. This is a typo right here. So our cell has a cell membrane or a plasma membrane. Our nucleus has a nuclear membrane. Our endoplasmic reticulum has its own membrane, and there are membranes of the mitochondria, the lysosomes, and the Golgi apparatus. Now let us put our focus on the cell membrane or the plasma membrane. The cell membrane envelopes the cell, and this is composed mainly of proteins and lipids. This separates the ICF within the cell from the ECF outside of the cell. Lipid-soluble substances can penetrate this lipid bilayer. So the basic structure of the cell membrane is a lipid bilayer. Okay, so this is the structure of our cell membrane. Now let's talk about the lipid bilayer of the cell membrane. Again, the basic structure of our cell membrane is the lipid bilayer. Now, why is this important? This is important because it allows for the selective permeability of the cell. And this is composed of your phospholipids, your sphingolipids, and your cholesterol. Now, your sphingolipids are for the protection of the cell from harmful environmental factors, for signal transmission, and for adhesion sites for extracellular proteins. And your cholesterol, these controls the fluidity of the membrane, and they also determine the degree of permeability to water-soluble substances. Now let's talk about the phospholipids. So the phospholipids are the most abundant with a head and a tail. So if we look at the picture right here, the one encircled in yellow, those are the heads, and the one in between are the tails. Okay, so the head of the phospholipid is attracted to water. So if we draw it the same way, this is your head, okay, and it'll have two tails, okay, that are together. Now the head again is attracted to water, so it is hydrophilic, so it loves water, okay? And we have a tail that repels water or is hydrophobic. I hate water. Okay, so again, the head is hydrophilic, which means that it is attracted to water, while the tail repels water or is hydrophobic. So there are water-soluble substances and fat-soluble substances. So your water-soluble substances are different ions. We have glucose, and we have urea. Now, you also have fat-soluble substances, which are your oxygen, your carbon dioxide, and your alcohols. So remember a while ago that we talked about the lipid bilayer, and that lipid-soluble substances can penetrate this lipid bilayer, meaning that fat-soluble substances can penetrate the lipid bilayer of the cell membrane. Now again, looking at the picture to the right, you can see there are a lot of attachments to it. These attachments are proteins, and there are several types of proteins with different functions. So as you can see right here, this one is an integral protein, and we have a peripheral protein. So these attachments are your cell membrane proteins. Again, we have two. We have our integral proteins and our peripheral proteins. Now, our integral proteins, we have three different types. So they can work as channels, they can work as carriers, they can work as receptors. So these proteins are mainly glycoproteins that are embedded in the membrane, and they provide selective permeability to substances that diffuse between the ECF and the ICF. Integral proteins as channels. So as channels, they provide structural pores that allows the free movement of water molecules and water-soluble substances to diffuse in and out of the cell. As carriers, they are transporters. They transport substances that cannot penetrate the lipid bilayer. An example where we can use this is during active transport, which we will talk about further soon. So active transport is a movement from lower to higher concentration where they use integral proteins as carriers. Now, we also have integral proteins as receptors. So these are used for water-soluble chemicals that cannot easily penetrate the cell membrane. So an example would be where ligands would bind to the receptors, and the proteins in the cytoplasm become messengers and relay the signal from the receptors outside to the interior of the cell. And finally, we have here our peripheral proteins. So they function similarly as enzymes or controllers of transport of substances through the cell membrane pores. Okay, again, this is enzymes. So they function the same way as enzymes, and they control the transport of substances through the cell membrane pores. So let's now move forward to the cytoplasm and its organelles. First, we have the cytosol. So the cytosol, this is the jelly-like fluid portion of the cytoplasm. This is where organelles are dispersed, and they contain mostly of dissolved proteins, electrolytes, and glucose. Let's move forward to our endoplasmic reticulum. Our endoplasmic reticulum aids in transporting and processing molecules made by the cell. Alright, so if you look at the picture here on the right, this is your endoplasmic reticulum. These things right here, and you can see that some parts have those dots that makes the surface kind of rough, and there are also smooth areas. So we have rough or granular endoplasmic reticulum, and we also have a smooth or a granular endoplasmic reticulum. Now, what are those rough parts? Those rough parts are called your ribosomes, and those are located in your rough endoplasmic reticulum. Now, your ribosomes are a mixture of RNA and proteins, and they are important for protein synthesis. Now let's talk about our Golgi apparatus. The Golgi apparatus works with the endoplasmic reticulum and receives transport vesicles or endoplasmic reticulum vesicles. They transport substances that are processed to form lysosomes and secretory vesicles, and they are essentially the packaging house of the cell. Now, we have lysosomes. Our lysosomes are the intracellular digestive system. They break off from the Golgi apparatus and they digest or engulf foreign or unwanted material. They also contain high concentrations of hydrolytic enzymes that are capable of splitting organic compounds. And then we have our peroxisomes. Peroxisomes are formed by self-replication or comes from the endoplasmic reticulum instead of the Golgi apparatus. They contain oxidases rather than hydrolases, because remember, the lysosomes, they have hydrolytic enzymes. Now, our peroxisomes have oxidases. So they oxidize poisonous substances in the cell and they catabolize long-chain fatty acids. The next one here is our mitochondria, which is the powerhouse of the cell. They produce ATP. If you look at the picture on the right, they would have a cristae because the cavity of the mitochondrion has a matrix that has large quantities of dissolved enzymes that are necessary for creating energy. So that is why the cristae are formed, okay? Because what's inside the cavity of the mitochondria is so extensive. And those oxidative enzymes that are found in between those shelves or the cavities, they allow for the oxidation of nutrients that form carbon dioxide and water and liberated energy, or our ATP. Then we have our nucleus. Our nucleus is the control center of the cell. They contain large quantities of DNA. So our DNA comprises of the genes. The genes will then determine the characteristics of the cell protein. Now this is something that is good to know. So there are 46 chromosomes, meaning there are 23 pairs, and this happens via mitosis. So mitosis is for cell reproduction, for growth, and for replacement of worn out cells, where two identical sets of genes split to form two daughter cells. Another thing that's good to know, if you're wondering, we also have meiosis. Meiosis is for the production of egg and sperm cells. So these are for our sex cells. Now they come from haploid. They create a haploid, meaning from a single cell with a single set of chromosomes, leading to 4 daughter cells. And then we have our nuclear membrane, which again separates the nucleus from the cytoplasm. And finally, the nucleoli, which is an accumulation of large amounts of RNA and proteins within the nucleus. All right, let's now talk about the transport of substances through the cell membrane. Blood circulates in the body through our blood vessels, and the fluid movement between blood capillaries and intercellular spaces happens through diffusion. So, the structure of capillaries are permeable to most molecules in the plasma of the blood, except for plasma proteins, which are too large to readily pass through the capillaries. So, we were talking about this picture right here. Okay, the exchange of nutrients between the capillaries and intercellular spaces is via diffusion, which is a net movement of ions from regions of higher to lower concentration. So again, remember that diffusion is a movement of ions from higher to lower concentration. All right, now let's look at our transport systems. The first part is our passive transport, and we'll talk about active transport later on. So for passive transport, we have diffusion and osmosis. In diffusion, we have simple diffusion through a lipid bilayer, and then we have facilitated diffusion through carrier proteins. In facilitated diffusion, we have non-gated ion channels, we have voltage-gated ion channels, and we have ligand-gated ion channels. Alrighty, let's first talk about simple diffusion through the lipid bilayer. So again, our cell membrane has a lipid bilayer in which there are kinetic movement of molecules or ions through this membrane opening without any interaction with carrier proteins. Hence, it's called simple, because these molecules or these substances can just easily go through the cell membrane without using any carrier protein. So, if we can remember, what are those substances? So, those substances are your lipid-soluble substances. So, these are your lipid or your fat-soluble substances. What are those substances? We have our oxygen, we have our nitrogen, we have our carbon dioxide, and we have alcohol. So these substances can just easily go through the lipid bilayer without any problem. Hence, it's called simple diffusion. Let's now move forward to simple diffusion, but now through protein channels. So when we are using protein channels, there is a transport protein in the membrane that facilitates diffusion of example water across the membrane made possible by a protein channel. If we look at this blue thing right here, this big thing right here, it is just there in the cell membrane. It's called a channel, and that's where water can just easily pass through, in and out. Now for non-gated ion channels, these are just open tubes from the cell membrane that allows the steady flow of ions continuously. So it is selectively permeable depending on the diameter of the pore and the electrical charges needed to pass through. So these channels are just present in your cell membrane, and they allow the continuous flow or the easy flow of some ions depending on the makeup of that channel protein. Next, we have simple diffusion through voltage-gated ion channels. So why is it called voltage-gated? This is a gating that allows for controlling ion permeability of channels that may be open or closed and are regulated by electrical signals. Okay, that's why it's called voltage gated. So an example here with the picture on the right, we have K gates and NA gates that play a role in action potential. So what we use here are electrical charges. Next, we have simple diffusion through ligand-gated ion channels. So they use chemicals. So if voltage-gated ion channels use electrical signals, ligand-gated ion channels use chemicals. So an example are hormones, second messengers, or neurotransmitters that bind to the channel that opens or closes the gate. So an example here, if you look at the picture on the right, this channel is closed, and when this neurotransmitter right here attached to the receptor, it opened the gate. So that is an example of a ligand-gated ion channel. So this neurotransmitter right here is called your acetylcholine. This is important for the transmission of nerve signals from one cell to another and from nerve cells to muscle cells to cause muscle contraction, which we will be talking about further on. Alrighty, now let's talk about facilitated diffusion. We're done with simple diffusion. We're now going to talk about facilitated diffusion. Facilitated diffusion, that's why it's called facilitated, because it requires interaction with a carrier protein. So if simple diffusion does not need any interaction with carrier proteins, facilitated diffusion requires interaction with a carrier protein. So an example here is, so it's essentially called a carrier-mediated diffusion. Now, if you look at to the right, we would have this weird-looking graph right here. So the rate of diffusion is faster than simple diffusion and approaches a maximum called Vmax. Okay, so that's the difference between simple diffusion versus facilitated diffusion, is that yes, the rate of diffusion is faster, but if you look at facilitated diffusion here, it would reach a Vmax compared to simple diffusion. Okay? We will now talk about osmosis. So osmosis is the movement of water or solvent across a semi-permeable membrane from high to low solvent concentration. Diffusion is the movement of the molecules from high concentration to low concentration. Osmosis is the movement of the solvent. So in osmosis, only the solvent moves. So only the water moves. If we think about it, when we say high concentration of the solution, or when we say high water, there's high solvent, meaning it is high water. That also can mean that there is low solute. Now we also have a term here, osmotic pressure. So osmotic pressure, this is the amount of pressure required to stop osmosis. Why is this important? Because if there is no osmotic pressure, the water will just keep on moving and transferring and transferring. So osmotic pressure is important to establish equilibrium. Okay, so if there are higher concentration of a solution, there is a higher osmotic pressure. All right, so again, if osmotic pressure, if there is a higher concentration of the solution, there will also be higher osmotic pressure. So an example here, an example of osmosis is when our skin absorbs water and it bloats. So remember, think about it when you're swimming or you've been taking a bath for a long time. Your skin starts to bloat, and to compensate for that increased surface area, our skin wrinkles. So within that situation, osmosis is happening. Let's briefly talk about the different types of the following solutions. So we're going to compare the osmotic pressure of the following solutions. Let's first look at an isotonic solution. So an isotonic solution meaning there is the same concentration of the solute and same osmotic pressure. If you look at the picture on the right, this in a cellular level, this is how it may look like. So if there were 4 particles inside, there would also be 4 particles outside. Okay, so there is going to be an equal transport of water. All right, now let's look at a hypotonic solution. Hypotonic solution, right here, meaning there is lower solute, thus there is lower osmotic pressure. So if we draw that, there is going to be lower solute. So then where would the movement of water go? Again, the movement of water would go to higher concentration of water to lower concentration of water. So there is less water on the inside of the cell, therefore water will tend to move inside of the cell, thus causing this swollen structure. Now let's go to hypertonic solution. So hypertonic meaning there is greater solute. So if we draw that, there would be lesser here and a lot here on the outside. So what would happen then is water would tend to move out of the cell, thus causing that shrunken image. Okay, so that is your hypertonic solution where there is high solute, hence there is also high osmotic pressure. Okay. Alright, now we are done with passive transport, which is diffusion. Now we are going to move forward to active transport. So this right here is incorrect because diffusion is not an active transport. Okay? Alright, now our active transport, we have a primary active transport and we have a secondary active transport. So in our primary active transport, we have our NAK pump, and for our secondary active transport, we have our co-transport and our counter-transport. Now, active transport as opposed to diffusion, if we remember, diffusion is the movement from higher to lower concentration. Now, active transport is the movement from lower to higher concentration. We are now going against the concentration gradient. Now, to be able to go against the concentration gradient, we are going to use additional energy called ATP. An example of that energy is ATP. So this transport uses carrier proteins, the same as facilitated diffusion, but the carrier proteins in active transport is capable of imparting that energy, this energy right here, to the transported substance to move it against the gradient. Let's first talk about our primary active transport, which is our sodium-potassium pump. So this is a transport process that pumps sodium outward and potassium inward the cell membrane to establish a negative electrical voltage called our resting membrane potential. Okay, so this primary active transport, primary meaning the energy used is directly from the breakdown of ATP. So it is ATP direct. So it directly uses whatever is broken down from ATP. Okay, so if we look at the picture here, it would have 3 binding sites for Na or for sodium, and it would have 2 binding sites for potassium. 2 binding sites for potassium. So we can say that 3 sodium will go out and 2 potassium will go in. So this is how the sodium-potassium pump will happen. So what happens here is the sodium-potassium pump will bind 3 sodium ions and a molecule of ATP. Now the sodium-potassium pump will not work if there is no ATP. If you remove ATP, the protein, the channel protein here, will not work. It will not open the gates for potassium. That is why energy is needed. Once sodium binds, the splitting of ATP will provide the energy to change the shape of the channel. So the sodium ions are driven through the channel. So you can see the Na, the sodium ions are already inside the channel. So the sodium ions are then released to the outside of the membrane, and the new shape of the channel allows 2 potassium. So wala pang potassium dito. Now the potassium is here. So this will allow 2 potassium ions to bind. The release of the phosphate allows the channel to revert to its original form, releasing the potassium ions on the inside of the membrane. So once the phosphate here detaches from the channel, it's going to allow potassium to go inside of the channel or to go inside of the cell membrane. Alright, now let's move forward to secondary active transport. So for secondary active transport, the energy used here is derived from the energy that has been stored in the form of ionic concentration differences created originally by the primary active transport. So if our primary active transport used the energy directly from the breakdown of ATP, let's think about secondary active transport as the use of the energy from indirect breakdown of ATP. So we have 2 types here. We have the co-transport or the synport, and we have counter-transport or the antiport. In a very simple definition, co-transport, meaning the substances travel in the same direction, and counter-transport, meaning the substances will travel in the opposite direction. So in the picture here on the right, let's try to explain this. We have an excess sodium outside the cell from the sodium-potassium pump. Sodium will always try to attempt to diffuse inside, and sodium may pull other substances along. So bakit may excess sodium? It's because, again, 3 sodium goes out and 2 potassium goes in. So sodium may pull other substances along with it through a coupling mechanism. So this is usually used for transport of glucose and amino acids. So if you look here, this picture right here, so that's an example. You see dinala ng glucose yung dopamine and GABA papasok. Okay? And then we have our counter-transport or our antiport. So again, the substances will travel in the opposite direction. So now, sodium will attempt to diffuse to the interior of the cell because there is high sodium concentration outside. It would like to go, again, the movement is high to low concentration. So it would like to go to the lower sodium concentration inside. And the substance to be transported will come from the inside the cell to be transported outside. So an example here would be your sodium countertransport of calcium and H ions. Alright, so that is the end of the first part of our topic 1. If you need to go back to any part of the slide, please do so. If you need to take a break, then take a break. And when you're ready, we can now proceed to the next subtopic, which is the functional organization of the nervous system. Again, please take a pause, take time to read the learning objectives. When you're done, we are now going to proceed to our topic outline. So this is our topic outline. Again, you can pause this part of the slide. You can see and digest how we are going to discuss the topics within the functional organization of the nervous system. Alright, so let's now proceed. This is the anatomical division of the nervous system. So essentially, we have a peripheral nervous system and a central nervous system. Our central nervous system is made out of the brain and the spinal cord. And then we have, for the peripheral nervous system, we have our cranial nerves. So for our PNS, we have our cranial nerves and our spinal nerves. So why did I not point out this picture right here, the part here? Because this is the physiologic division or the functional division of the nervous system. So please refer to this. So the anatomical division, we have our central nervous system, which is your brain and your spinal cord, and then your PNS, it has your cranial nerves and your spinal nerves. Okay, so let's now talk about our central nervous system. First, the brain. Now, everything here that I will be saying, I will be mentioning a bit of the functions of each of these parts, but there's really no need to put so much focus on it now, because this topic actually is just to sort of like an introduction to help you guys understand how everything is organized, because each of these will be discussed more specifically as we go through the course. Okay, so let's start first with our cerebrum. Our cerebrum, or our forebrain, has a diencephalon and a telencephalon. Alright, so let's talk about the diencephalon. We have the thalamus. So the thalamus, this is the relay station this is a relay station of information, and it plays a role in sleep, in wakefulness, in consciousness, and also plays a role in learning and memory. So that is your thalamus. Next, we have our hypothalamus. Our hypothalamus plays a role in homeostasis and control of the endocrine system. Okay, so that is your diencephalon. Your diencephalon has your thalamus and hypothalamus. Next, let's talk about the telencephalon. The telencephalon has your cerebral cortex, which we all know is the higher level processes of the brain. So that is its main focus. Next, we have our subcortical white matter. Now, our subcortical white matter is essentially the interconnection of cortical and subcortical areas. And finally, we have our basal ganglia for our telencephalon. Our basal ganglia is important in motor control, in executive functions, in behavior, and emotions. Alright, so that's it for the cerebrum. Okay, next we have our cerebellum, or our hindbrain. We have two parts underneath the cerebellum. We have the cerebellar cortex and the cerebellar nuclei. And then we have the brainstem. So in a general perspective, the brainstem is used for the subconscious body functions. So that is its main role. Now we have our midbrain here, and we have our pons. The pons is important because it connects the brainstem to the cerebral cortex. So it is also the coordination center for signals and communications. And then we have our medulla oblongata. So this is a conduit or a channel for the ascending and descending nerve fibers, and it controls vital processes like our heartbeat, our breathing, and blood pressure. Alright, and then after the brain, the other part of the central nervous system is our spinal cord. Okay, now for our spinal cord, it is essentially made out of, we have a gray matter, So it's called gray matter because its color is gray. And it's made up of nerve cells where processing is done. And we have a white matter. So this one is a white matter because the color of it is white because of the myelination or the lipids that surrounds it. Now, this white matter consists of nerve fibers that serves as channels of communication. Okay? Alright, let's now proceed to our peripheral nervous system. So, our peripheral nervous system is composed of cranial nerves, spinal nerves, and ganglia. Now, for our cranial nerves, these are 12 pairs of nerves that exit the skull through foramina. So, foramina are openings in your skull, and of which these parts I hope will be discussed more in detail in your ANA 212 subject. And then we have your spinal nerves, which are 31 pairs naman. So if your cranial nerves are 12 pairs, your spinal nerves are 31 pairs that exit the vertebral column through the intervertebral foramina. So as you have seen in the previous slide, you saw the words thoracic, lumbar, all those parts. And there's also coccygeal. So it would have 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. So if you add all of those together, it's going to be 31 pairs. Alright? And then we have ganglia. So what is a ganglia? A ganglia, these are masses of nerve tissue. So these are masses of nerve tissue. Nagkumpul-kumpul na nerve tissue. So we have a sensory ganglia or our posterior root ganglia and also an autonomic ganglia. These are the efferent nerve fibers of the ANS. So efferent nerve fibers of the ANS. Now we are going to talk about the functional divisions of the nervous system as mentioned previously. So our functional division, it would just be divided between somatic and autonomic nervous system. So for our somatic nervous system, this innervates the structures of the body wall. So it has sensory input and it would have a motor output. For our ANS innervates involuntary structures such as your heart, your smooth muscles, or your organs and glands. Our ANS has sympathetic. Sympathetic prepares the body for emergencies. And your parasympathetic, these are for conserving and restoring energy. We are now done discussing the divisions of the nervous system. Now it's time to talk about the cells of the nervous system. There are 2 major cells of the nervous system. One is our neuron and the other is our neuroglial cells. Alright, so an overview, if you look at the picture below, there are also different cells for your PNS and different cells for your CNS. Okay, so if your PNS has satellite cells and Schwann cells, your central nervous system would have, again, the neurons and neuroglial cells. Okay, let's first talk about our neuroglia. Our neuroglia are non-excitable neurons that support the nervous system. They do, however, outnumber neurons, but they do not form synapses. So neuroglia, they are non-excitable and they do not form synapses, but they play an important role in myelin formation, in the guidance of developing neurons, in the maintenance of potassium levels in the ECF, and in the reuptake of transmitters after synaptic activity. Okay, our neuroglia has different types. We have our macroglia, we have our microglia, and our ependyma. For our macroglia, we have 2. We see here our astrocytes and our oligodendrocytes. Now, the astrocytes are termed astrocytes because they have a star-like appearance, okay? And they are found in the gray matter. So they regulate neurons and surrounding environment by secreting growth factors. And they repair through reactive gliosis. So if we talk about astrocytes, we think of the word repair. These are the repairmen of our nervous system. Now, let's talk about the oligodendrocytes. This is our oligodendrocytes. Our oligodendrocytes are found within the white matter, and they myelinate nerve fibers in the CNS. So when we think of oligodendrocytes, we think of myelination. Okay? Alright, now we have also our microglia right here. These are our microglia. Now, our microglia are macrophage-like cells, and they are the soldiers of the nervous system. They function similarly to white blood cells. So when we think of our microglia, we think about soldiers. They are the protectors, okay? They are like macrophages. And then we have our ependyma. We have our ependymocytes, which are important for our CSF. So our ependymocytes, they circulate CSF, and they also absorb CSF. Okay? Our tanycytes transport substances from the CSF to the hypophyseal portal system, and our choroidal epithelial cells produce and secrete CSF. So these guys right here, they produce and secrete our CSF. Let's now proceed to the other cell of the nervous system, which are our neurons. Now, our neurons are the excitable cells that are specialized for the reception of stimuli and conduction of nerve impulse. So if you remember, our neuroglia are non-excitable. Our neurons are excitable cells. Okay, so the difference between the other cells from the body is that it has dendrites and axons, which are long processes used for the communication between other neurons, and they do not undergo division and replication. Now we have our upper motor neurons and our lower motor neurons. So our upper motor neurons, they originate from, that's the difference, they originate from the brain, and they go down to either the brainstem or the spinal cord. Now for our lower motor neurons, they begin in the spinal cord and go to innervate muscles and glands throughout the body. Okay, so upper motor neurons from the brain to the brainstem or the spinal cord. Lower motor neurons, spinal cord to the muscles or glands. Let's now talk about the parts of a neuron. So we have the cell body or the soma. So these are the metabolic and genetic center of a neuron. This is also the receptive pole of the neuron. Receptive meaning it receives things, it receives impulses, it receives information. Okay, so that's our cell body. This right here is our cell body. Okay, next we have our axon. Our axon right here, this conducts impulses or action potentials away from the body. Okay, so axon is away from the body. And then we have our dendrites. Now our dendrites right here, all of these are dendrites. These are branches of neurons that extend from the cell body, and they receive information, or they receive incoming synaptic information. All right, and then we have the myelin sheath, which serves as insulators. Okay, so why is a myelin sheath important? Myelinated nerve fibers are stimulated only at the nodes of Ranvier. So if we look at the picture right here, we have nodes of Ranvier. These are areas where the myelin is absent. So if you see the yellow parts here, those are parts of the axons that are myelinated, and the ones in between are the nodes of Ranvier. So later on, we will be talking about action potentials, but just a brief introduction, there will be leaping of an action potential from one node to another, and that is called our saltatory conduction. Okay? That is called our saltatory conduction. Now, this leaping is a way for the information to go through faster. Okay? So this is a more rapid mechanism found compared to our non-myelinated fibers. So examples are for motor functions, vibration sense, directional touch. Okay? And our neuron's cell body is like that of the other cells. So the plasma membrane of a neuron is also the site for the initiation and conduction of nerve impulses. Alright, if our neuroglial cells have types, our neurons also have different types. So there are three major categories. We have neurons according to the number of processes extending from the soma, neurons according to size, and neurons according to function. Now, for our neurons according to number of processes extending from the soma, we have unipolar. So our unipolar neuron, these are found in our dorsal root ganglia. Okay? And we have our bipolar neurons. So our bipolar neurons are found in our sensory systems. Okay? So they are found in our retina, in our cochlea, in our vestibular ganglia. And then we have our multipolar neurons that are found in fiber tracts. So these are found in tracts of the brain to the spinal cord. Okay, so those are the types of neurons according to the extensions from the soma. All right, now let's talk about neurons according to size. We have Golgi type 1 and Golgi type 2. So our Golgi type 1, these are long axons. So they have long axons. Okay, and they form fiber tracts of the brain and the spinal cord. So examples of these are the pyramidal cells of the cerebral cortex, Purkinje cells of the cerebellar cortex, and motor cells of the spinal cord. Now we have our Golgi type 2, which are smaller than your Golgi type 1. So they have short axons, and they outnumber Golgi type 1. They also have shorter dendrites, and they are found in the cerebral and cerebellar cortex. Okay, and then we have according to function. So we have sensory neurons and we have motor neurons. Now, sensory neurons, these are afferent. Okay, afferent. They carry information from sensory receptors towards the CNS. So, for example, for our trunk and extremities, we have our dorsal root ganglion. For our face, we have the ganglia adjacent to the brainstem or the nuclei within the brainstem. And then we have our motor neurons. Our motor neurons are efferent. Okay, meaning they carry the motor information away from the nervous system to the muscles and glands to initiate an action. Okay, so sensory neurons are afferent. They carry information towards the CNS. And your motor neurons are efferent. They carry it away. Now let's talk about synapses. So neurons and neurons connect with each other, and in between those connections, they will form synapses. Transmission of information between neurons occurs at synapses or at synaptic junctions. So impulse transmission involves the release of a chemical transmitter substance, and the current passes directly from cell to cell through this specialized junction. So this is a picture of a neuron and a neuron connecting with each other. And if we zoom in further, this is what it's gonna look like. So that is our synapse. Now, we have a presynaptic cell. Okay, so this is the presynaptic terminal. And the one after that is your postsynaptic terminal or your postsynaptic cell. Okay? And in between, that is your synaptic cleft. Okay. there are different types of synapses. We have chemical synapses and electrical synapses. So sometimes there is no need for the use of neurotransmitters or these chemicals. So that is where your electrical synapse comes in. So we are going to categorize them in these parts. We have a synaptic cleft, we have the mediators, we have the synaptic delay, transmission of impulse, and examples. So for our electrical synapse, is there a synaptic cleft? There is no synaptic cleft, but there is a gap junction. For chemical synapses, you can find a synaptic cleft. Now, for the mediators of our electrical synapses, we use ions. Now, for our chemical synapses, we use chemicals or our neurotransmitters. For synaptic delay, there is not much delay with your electrical synapse. For our chemical synapse, there is a significant synaptic delay. For the transmission of impulse, it is bidirectional, so it can go away and then it can go back. For our chemical synapse, it is only unidirectional. Okay, examples where electrical synapses are used are found in your retina, in your olfactory bulb. Now for your chemical synapse, examples are found in most of neuron junctions and in muscle contraction. Alright, now we're on to the last slide of our subtopic 2. This is our neuronal groupings and connections. So essentially we have two. We have the nucleus and the ganglia. So this nucleus is different from the nucleus of a cell. So this nucleus is used as a term for clusters of nerve cells that are found in the CNS. Okay? Now your ganglia are clusters of nerve cells that are found in your PNS. So an example for your nucleus are your caudate, your putamen, your dentate, your emboliform, or your subthalamic nuclei. Now your ganglia, these again are clustered nerve cells in your PNS. So an example of this is your dorsal root ganglia. Another example are your autonomic ganglia and your cranial nerve ganglia. So these clusters of nerve cells sometimes connect further by bundles of axons, and they form either tracts or fasciculi. Okay? And when those tracts combine together and aggregate together, sometimes they form columns. So you might see this term columns or funiculi. So these are aggregates of tracts in the spinal cord. Now you may also see the term lemnisci. Okay? Now these naman are tracts within the brain. Okay, so those are our neuronal groupings and connections. And that is also the end of our subtopic 2. On to the next, we have our membrane physiology, nerve and muscle action potential. So again, please pause and read our learning objectives. And when you're ready, we're going to go to our topic outline. So this is our topic outline, and I need you guys to see what we are going to discuss. So again, pause it, and once you are done digesting the information here and seeing the whole process, we're going to go ahead and start with the lecture. Let's start with the resting membrane potential, which I have mentioned briefly a while ago. So our resting membrane potential is the electrical charge inside a cell when nerve fibers are not transmitting signals. Okay, so this is their steady state. The resting membrane potential of large nerve fibers when not transmitting nerve signals is about negative 70 millivolts or negative 90 millivolts. So these numbers right here are the ones that are usually used, and they can sometimes be board sensitive, but it's for now we're not really going to focus on the boards yet. So this is just something that's good to know. So for the nerves, it's negative 70 millivolts. For our muscles, it's negative 90 millivolts, and for smooth muscles, it's negative 50 to 60 millivolts, or to negative 60 millivolts. We're now going to talk about the mechanisms that generate our resting membrane potential, which we all have talked about at the start of the lesson. So we have, again, let's welcome back our passive transport and our active transport. So remember, for passive transport, there is movement through a concentration gradient, and we also have our potassium leak channels through our channel proteins. So if you remember, our channel proteins looked like this one right here, okay? And then we have our active transport, which is our NAK pump, or our sodium-potassium pump. So all cell membranes of the body have a powerful sodium-potassium pump. More Na goes out, or more sodium goes out. Remember, 3 Na out. We have 3 Na going out, and we have 2 potassium going in. So less in of potassium, and this causes a negative potential inside the cell membrane because more sodium goes out and less potassium goes in. Now we're going to talk about our electrical communication. So we have our local potentials and our action potentials. So in hindsight, we have our membrane potentials, right? Okay, so when our membrane potential is at rest, that is our resting membrane potential. Okay, now when something is stimulated, so when it's under stimulation, we now have these two. If it only reaches the subthreshold, that's going to be your local potential. Now, if something reaches the threshold, okay, that's going to be your action potential. Now let's talk about our local potentials. So our local potentials, these are a way for neurons to communicate, but they dissipate in space and time. So these are transient change of membrane potential in a localized area of the membrane. So I need you guys to remember this. When something is hyperpolarized, it means it has become more negative. And when we depolarize something, it is more positive. Okay, so again, if we hyperpolarize something, it is more negative. And when we depolarize, it is more positive. Okay, so summation can happen in our local potential. So rated potentials, meaning the larger the stimulus, either a stretch or a pressure, the larger the depolarization, and it is also additive. So an example of this would be your muscle stretch receptors, your touch pressure receptors, or your Pacinian corpuscles. Next, we have our action potentials. Our action potentials, they occur as a result of local potentials, and they are all or none responses. All or none meaning either you trigger an action potential or you don't. It's like flipping a switch. So a neuron will always send the same size of action potential. Action potentials are also self-regenerative, so the electrical signals propagate throughout a neuron along its axons. Neurons can generate action potentials because they contain specialized sodium channels that respond to depolarization and changes in the resting membrane potential. We're now going to talk about the stages in action potential. So we have the resting potential. Next, depolarization will happen, and then repolarization, and then we have hyperpolarization. So that is the general stages in the generation of action potentials. Okay, so let's go through the stages. So again, the resting potential is maintained by our NAK pump, or our sodium-potassium pump. So this is essentially how our sodium-potassium pump happens, okay? So what happens to generate an action potential is it always starts with a depolarization. So that happens at negative 55 millivolts. So at negative 55 millivolts, sodium channels or sodium gates open and they will start to depolarize. So when we say something is being depolarized, remember that it becomes more positive. So as sodium ions rush in, the inside of the cell becomes more positive. Okay, now an action potential will reach its peak and the potassium gates will start to open. At this point, when we reach the peak, our potassium gates open. Okay? At this point, there is what we call an absolute refractory period. Now, with an absolute refractory period, that means it's absolute. You cannot produce another action potential. At this point, you cannot produce another action potentials. So, potassium gates are open. Potassium will now go out of the cell. Okay, now at this point, our sodium gates cannot open. Habang nagre-repolarize tayo, which means mas nagiging negative tayo, hindi na naka-open yung sodium gates natin. So, wala nang pumapasok na sodium. Alright? Now, as it becomes more negative, remember, hyperpolarization, mas nagiging negative yung sa loob ng cell. Yung potassium channels natin, it remains open, causing further potassium efflux. So at this point, there is what we call a relative refractory period. Meaning, it would need more depolarization to produce another action potential. Relative refractory period, meaning pwede pang magkaroon ng isa pang action potential. So, for example, at rest. Okay, at rest, meron tayong negative 70 millivolts. And then the threshold to incite an action potential is negative 55 millivolts. So that is around 15 millivolts worth of depolarization. Okay, but let's say we are at a hyperpolarized state. Nandito tayo. So we are at, let's say, negative 80 millivolts. Papapuntahin natin para maabot yung threshold na negative 55. So that would be around 25 millivolts worth of depolarization. So that is your relative refractory period na sinasabi na pwede pang magkaroon ng isa pang action potential but you would need more depolarization. Alright, so let's try to further visualize pa our action potential. Okay, so remember again, an action potential will always start with depolarization. Okay? So during this stage, alright, mabibreak natin yung resting membrane potential kasi kapag na-reach na yung kung saan magdi-depolarize, which is at the threshold is at negative 55 millivolts, dyan na mag-start mag-open yung sodium channels natin. So what's going to happen is sodium will go, it will rush in, making the inside more positive. So again, it's depolarization. So nagsa-start tayo from negative 70 millivolts to negative 55 millivolts. Dito na nag-open yung sodium channels. Aangat pa yan hanggang maging positive. Okay, pag umabot na tayo sa positive 35 millivolts, when we get to 35 millivolts, what will happen now? So, ang mangyayari, magbubukas naman yung potassium channels natin. So, yung potassium na nasa loob, okay, lalabas yan. Alright? So, ang mangyayari ngayon ulit, magsa-start na ulit maging negative yung loob. Kasi, at that point, closed na rin yung sodium channel. So, wala nang pumapasok na positive. And then, meron pa tayong lumalabas na positive potassium ions. So what's going to happen here is nagiging negative na yung membrane ulit on the inside. Okay? Hanggang sa mas lumabas ng lumabas yung potassium, mas magiging negative pa yan. Hanggang umabot tayo sa hyperpolarized state. Again, we have our hyperpolarized state. Okay? So it's around, let's say, an example is negative 80 millivolts. So that is below our negative 70 millivolts. Alright? So then, when we are at the hyperpolarized state, again, it becomes more negative. Then, yes, our potassium ions will still continue to go outside until when we are going back to our resting membrane potential, our K channel will start to close. Alright, but there are our rectifier, okay, our inward rectifier for potassium channels, okay, that will allow some potassium to go in, and that will bring it back to our resting membrane potential. So this is the summary for the stages in our action potential. So again, an action potential will start with depolarization. So our sodium channels will open. Okay, so the ion sodium will go inside, making the inside more positive. And then we will reach our repolarization stage where sodium channels will now be closed and our potassium channels will fully open. So potassium ions will go outside, making the inside of the membrane negative. As it becomes more negative, we reach our hyperpolarization stage. So at this point, our potassium channels are still open and the inside becomes more negative. So our potassium goes outside and the inside becomes more negative, and then we return to our resting membrane potential as our potassium channel starts to close. Okay, at this point, potassium will still normally go outside as it always does and makes the inside of the cell or the membrane negative, thus achieving that resting membrane potential again. Alright, now we are going to proceed with the mechanism of muscle contraction. So this is basically what we will be talking about at this part. Please bear with me, it might be a little bit too overwhelming, so take breaks in between and just try to digest one by one, okay? Alright, we're gonna start easy first with the physiological anatomy of skeletal muscle. So this is how everything looks like. This is the organization of our skeletal muscle. So our muscle will become, so this is a whole muscle right here. This is the entire muscle right there. Alright, now within that entire muscle, we have our fasciculus. So this is our fascicle. This whole thing right here, that's your fascicle. Now within that fascicle, we have a fiber. Okay, so this is a muscle fiber. This thing right here. Now, within that muscle fiber, we have our myofibrils. Okay, our myofibrils have thin actin and thick myosin. And finally, we have your myofilaments. Okay, so that is the general organization of our muscles. So I mentioned actin and myosin. So our actin and myosin, these are protein molecules that are basically responsible for muscle contraction. So we're going to be talking about them a lot. All right, let's start with our myosin filaments. So our myosin filaments have A bands. Okay, this right here, that is your A band. Okay, now that A band is found in your myosin. So your myosin has protruding arms and heads, and they are called cross bridges. Okay. Next, we have our actin filaments that have I bands. This right here, this whole thing right here. your I bands are again found in actin, okay? So the ends of actin filaments are attached to a Z disc, and they extend to myosin filament. So this is your Z disc right here, and they attach also to your myosin filament. So everything is connected together. Now, when a Z disc and a Z disc connect together, that is called your sarcomere. Okay? So your actin, when we look at it further, it's going to be composed of your actin, your tropomyosin, and your troponin. Okay, so again, we have our myosin filament right here, which has arms and heads and are called cross bridges. Okay, so this is what the picture is showing. So it has arms and it has heads. All right. Okay, so going back to our actin. So this is what actin looks like. And the whole picture, when we combine your myosin and your actin together, this is how it looks like. Okay? All right. So our actin again has actin, tropomyosin, and troponin. So it has ADP molecules, and they are the active sites of actin filaments, which the cross bridges of myosin filaments interact with. Okay, so we have our tropomyosin right here. Okay, so our tropomyosin lies on top of the active sites of actin strands to block actin from the myosin. Okay, and then we have our troponin complex. Now, it's a complex because we have troponin I, it's three of them, troponin T and troponin C. So troponin, these are loosely bound protein subunits that initiate the contraction process. So troponin I has a strong affinity for actin, troponin T for tropomyosin, and troponin C for calcium ions. Now, in between myofibrils, so this in between, there are several of these, you will find what we call in the intracellular fluid our sarcoplasm with large quantities of potassium, magnesium, phosphate, and protein enzymes. It also has the important sarcoplasmic reticulum, okay? That is important in regulating calcium storage, release, and reuptake for muscle contraction. So we're going to be talking about calcium a lot because this ion is very important in muscle contraction. Right, now that we have had an overview of the organization of our muscles, we're now going to talk about neuromuscular transmission, excitation, contraction, coupling. Let's first proceed with sliding filament theory. So the sliding filament theory is a suggested mechanism of contraction of striated muscles, actin and myosin to be precise. So it says that they overlap each other, resulting in the shortening of the muscle fiber length. Okay, so you can see that in the pictures, right? So there is a picture that is showing a muscle at rest and then a muscle when it is contracted. Okay? Okay, so mentioned before also that in our sarcomere, we have our I band and our A band, and the one in between is your H zone. So your I band is in your actin or thin filament, and your A band is found in your thick filaments or your myosin filaments. Now, what happens to these bands when a muscle contraction occurs? So let's look at the picture again on the right. So when a muscle contraction happens, let's look at the A band first. Okay, this is our A band right here. As you can see, ang A band walang nangyari. It remained the same. Our A band alang nangyari. Nothing happened. It remained the same during contraction. Now let's look at our H zone, the one in the middle. Okay, the H zone, as you can see, disappears. Okay, so our H zone gone or disappear. And finally, let's look at our I band. Yung I band natin, here is our I band. Our I band. makikita natin na it shortens. So, I band shortens. Or, I ikli. Okay? It shortened. Now that we have talked about how muscle contraction happens generally, like through the sliding filament theory, we've talked about the different parts in the organization of a muscle, and previously we've talked about action potential, all these ions, all these transport systems. We're now going to look at how muscle contraction happens in a micro level. Okay? So it might get a bit too much again, a bit too overwhelming with all the information, with all the processes and the steps happening. So take time to pause and digest everything slowly at your own pace. Alright, so previously we've talked about synapses before, but now we're going back to a junction again, but this time more specific to a nerve and a connection of it to a muscle, and that is called your neuromuscular junction. So this is the synaptic connection between the terminal end of a motor nerve and a muscle. So in the axon terminal, there are many mitochondria that supply ATP that is used for the synthesis of an excitatory transmitter that is called your acetylcholine, which is very important in muscle contraction. So acetylcholine is our main neurotransmitter. Okay, so bago tayo magkaroon ng muscle contraction, merong nangyayaring events between the neuromuscular junction. Okay, so in hindsight, this is what happens when there is a muscle contraction. So again, it will always start with an action potential. An action potential will trigger the excitation-contraction coupling, magkakaroon ng power stroke, and then magkakaroon ng muscle contraction. After that, the cross bridge will release, and then magkakaroon ng muscle relaxation. So when we talk about it very simply, ganun siya nangyayari. Pero bago mangyari yun, titignan muna natin ano ba yung nangyayari sa NMJ natin. So what happens here is that an action potential will arrive at your presynaptic terminal. Okay, when an action potential arrives, the voltage-gated calcium channels in the presynaptic membrane, so there are calcium here, will open and calcium ions will enter the presynaptic terminal. So this will initiate the release of acetylcholine. This is our main neurotransmitter from the synaptic vesicle. So ACH will be released. Okay, now ACH will then bind to the ligand-gated sodium channels sa postsynaptic membrane. So itong ACH natin, magta-travel to, pupunta na sa NMJ natin, pupunta na sa other side, magbabind na siya sa receptors. Okay? Magbabind na yung ACH natin sa receptors. Now, when that happens, okay, the ligand-gated sodium channels open and sodium will enter the postsynaptic cell, causing the postsynaptic membrane to depolarize. Okay? When that happens, at lumagpas sa threshold yung depolarization, nagkakaroon naman ngayon ng action potential sa postsynaptic membrane natin. After na magkaroon ng action potential sa postsynaptic membrane na kailangan natin para magkaroon ng muscle contraction, itong mga ACH na nagbind sa receptors, aalis yan sa receptors natin. Okay? They will unbind from the ligand-gated sodium channels. And what happens next is magkakaroon ng release of what we call acetylcholinesterase. So mahabang word siya. Okay, so let's just write it down here. So acetylcholinesterase. So acetylcholinesterase is important para ma-break down niya yung ACH into acetic acid, okay, and into choline. Okay, it needs to break down ACH because if it does not break down ACH, our acetylcholine will continue to bind and bind and bind sa receptors, and it will continue to produce and produce action potential. So, we need to stop that. Otherwise, masyado ng maraming action potential. At yung nagbe-break down ng ACH natin ay our acetylcholinesterase. Okay? Once nagkaroon na ng breakdown of ACH into choline, okay, nag-breakdown na siya into choline and acetic acid, nagkakaroon ng reuptake of choline sa presynaptic membrane, alright? And yung acetic acid natin, nagdi-diffuse away from the synaptic cleft. Okay, so acetylcholine synthesis from choline recycled from synaptic cleft and acetic acid generated from metabolism in the presynaptic cell. Okay, so yun, etong lahat ng to ang nangyayari sa ating neuromuscular junction para mapaabot natin yung action potential sa postsynaptic membrane at madala natin siya sa ating muscles. Okay? Okay, now that we have talked about what happens at the neuromuscular junction and we now have brought the action potential to the postsynaptic membrane, the action potential now that we have will cause the depolarization of the transverse tubules. Now, these transverse tubules are found in your sarcolemma or your sarcolemma. And within that is your sarcoplasmic reticulum, again, which stores your calcium ions that are very important in muscle contraction. Now, when calcium is released, it will bind to troponin C. Okay, so calcium has been released, it's going to bind to troponin C. When troponin C has calcium, it will activate troponin I, and troponin I will inhibit the covering of actin, which is our tropomyosin. So the covering of our actin is our tropomyosin. Now, when that covering is out, it is going to expose the active sites of our actin. Now, contraction requires energy. So we have an ATP here that will enter the scene, and that ATP will be broken down into ADP and inorganic phosphate. Now, this event will cause the formation of cross bridges. So the myosin head will now attach to the active sites. Okay, so we now have the myosin head that formed a cross bridge with our actin. Okay, so connected na yung myosin at yung actin natin. Ang mangyayari na ngayon ay yung head ng myosin, it will cause a power stroke with the energy that it has. And that will pull the actin filament inward. Okay, hihilahin niya yung actin filament. Papasok. Alright? So during the power stroke, the myosin head, it's going to bend. See here? Nagbend at the hinge. And the ADP and phosphate are then released. Eto na sila. So the actin filament has now been pulled forward and the energy has been released from the myosin head. So then, the new ATP now, right there, attaches to the head, detaching it from the active sites. So again, ATPase activity will happen in the myosin heads. So energy will always be on the myosin heads. Alright, so myosin head now will return to its resting position and energy is just stored in the heads. So calcium then will go back to the sarcoplasmic reticulum via active transport, and active sites are being covered again in your actin filament, resulting to our muscle relaxation. Okay, so that is our excitation contraction coupling. So again, in hindsight, this is simply put, no? One, an action potential travels to the neuromuscular junction, and then the nerve will secrete acetylcholine. Then acetylcholine will promote excitation, and this will attach to the end plate and open the acetylcholine-gated channel. Now, sodium ions will diffuse the muscle fiber membrane. So, napunta na tayo sa postsynaptic part, and we have brought the action potential there. Now, the T-tubules and the sarcoplasmic reticulum, of which stores calcium, will depolarize, and it will release calcium. Now, once calcium has been released, it will bind to troponin C. Calcium, which is for calcium release and storage. Now, troponin C will activate troponin I, and troponin I will inhibit the covering of actin, which is our tropomyosin. Now, the heads will attach to the actin, causing a power stroke, which causes the muscle contraction. And then the cross bridge will end and the muscle will relax again. Alright, now that we have discussed how a muscle contraction happens, we're going to briefly discuss muscle twitch, tetany, and trep. Okay, so a muscle twitch happens when a single action potential will cause a brief contraction followed by relaxation. So it's just going to be a brief contraction and then relaxation. Okay, next we have tetanus. So tetanus happens with rapid and repeated stimulation. So contraction continues before complete relaxation happens. All right, so individual responses fuse into one continuous contraction. Now, there are two types of tetanus. We have complete tetanus, where there is no relaxation in between the stimuli, and there is incomplete tetanus, where there are periods of incomplete relaxation between summated stimuli. So if we look at complete tetanus here, there are no periods of relaxation as compared to our incomplete tetanus right here, where there are periods of relaxation. Okay? And then we have trep. So literally when you type trep in Google, you're gonna get pictures of staircases. So trep means staircase. Okay, so this is what we call a staircase phenomenon. So a series of stimuli is delivered to the skeletal muscle, and there will be an increase in tension during each twitch until after several contractions. So that stimuli will rise and rise and rise until magkaroon tayo ng uniform tension. Okay? So the strength of contraction remains the same or reaches a plateau. As you can see right here, this is the maximum tension for your trep versus the maximum tension in your tetanus. Okay, so this trep or staircase phenomenon is the basis for doing warm-ups. Alright, we are down to our last slide. Now, since we're already at the topic of muscle contraction, let's briefly talk about the different types of muscle contraction. Now, we have our isometric, isotonic, and isokinetic muscle contraction. So, isometric meaning there is no change in muscle length. So, isometric, you do not produce a movement here, okay? Because there are no changes in length during muscle contraction. Now, for isotonic, okay, there is shortening of the muscle. Now, there are two types of isotonic contractions. We have concentric contractions and we have eccentric contractions. Concentric contractions, the muscle shortens and this produces movement. And eccentric contraction, the muscle will lengthen and produces movement. So concentric shortens, eccentric lengthens, and both will produce movement. Okay, and then we have isokinetic, where the speed is constant during contraction, and this usually uses special machines. Okay, now if we look at the pictures available, these are some examples of our muscle contractions. So again, a concentric contraction is when a muscle shortens. So in here, when you are, think about it, when you are seated down and you're going to stand up, your quads will shorten, your quadricep muscles will shorten, and that is a concentric contraction. That is for you to be able to stand up. Now, when we are trying to control our movement as we sit down on the chair again, your quads will then lengthen again, and that will produce movement as you sit down. So that is what we call an eccentric contraction. The same thing when you lift a bottle, you shorten your biceps, And when you bring it down, you're still contracting your biceps, but you are lengthening it. So that is your eccentric contraction. We have now reached the end of our first topic. If you have any questions and if you didn't understand some parts of the lesson, please take note of them or put them down in our muddy points so we can discuss them together. Okay? Thank you very much for your attention, and I hope you learned a lot, or I just hope you got something out of this session. These are my references.